Thank you very much for the opportunity to present here at the BGRI meeting. As a student, that's quite a great honor. So I hope I'll be able to provide you with some interesting insights. I'll give you an overview of our work on the identification of likely airborne transmission routes of novel strains of stem rust. So in essence, what we try to do is quantify long-distance airborne dispersal in order to contribute to enhancing sampling strategies. So as an introductory example, let us look at current detection sites of the TTKSK rays. And then one of the immediate questions arising is, how does the pathogen spread? And in our work, we focus on airborne wind dispersal and specifically study questions like how often does it actually happen that pathogens get dispersed over these long distances? In which direction are they likely to travel? And what are typical amounts of spores being dispersed on regional and continental scales? So my presentation is structured along the following key questions. How do we approach the quantification of long-distance airborne routes? What are likely routes of transmission within and between South and East Africa, the Middle East, and South Asia? And finally, can we predict spore dispersal in real time? Now, I will be presenting the joint effort of this group of people here. We are working in a collaborative project with Dr. Laura Gilligan from the UK Meteorological Office, Dr. Dave Hodson from CIMMYT. I'm a PhD student in the Epidemiology and Modelling group of Professor Gilligan in Cambridge, and I'm continuing the work of two former group members, that is James Cox and Matt Hitchings. We are approaching the quantification of long-distance routes by combining large-scale meteorological datasets with high-performance computers in order to numerically study stem rust dispersal. Hereby, our modeling approach is quite straightforward. So we start with the host landscape. Here you see a map of the wheat-producing regions in our spatial domain of interest. And as I said, we're interested in quantification of key airborne transmission routes. So in order to be able to do that, we use large-scale meteorological datasets covering the entire domain of interest. So here you see the host landscape, and the idea is that we lay a three-dimensional grid of meteorological data on top of it, where each grid cell contains all the necessary variables for spore dispersal calculations. So for example, each grid cell contains mean wind values, mean wind direction, mean wind speed, as indicated by these arrows. We have these datasets which are available on a 3-hourly basis, and we use them in long-term studies over 10 years to identify key transmission trends. Now, to be more precise, we feed the meteorological data into an atmospheric dispersion modeling environment called NAME. NAME is a Lagrangian atmospheric dispersion model developed by the UK Meteorological Office, and we adapted to model uredinospore dispersal. Hereby, we consider key biophysical characteristics of uredinospores, and then we calculate turbulent atmospheric dispersion and consecutive dry and wet deposition of spores on wheat-growing regions in our domain of interest. Now, here it's essential to point out that we parameterize the model for the case of a local epidemic outbreak. So the results I'll be showing are valid for high inoculum load at the source, high spore numbers. The details of the model are beyond the scope of this talk, so let me show you a short animation illustrating our spore dispersal simulations. In this example, we release spores from a source location in Ethiopia, in the barley zone in Ethiopia. So the colors here indicate the height of the terrain, and so we fly into the Rift Valley from the north, and we see the Ethiopian mountains here. The black dots represent spores, and the gray areas sketch parts of the hourly deposition rate. Now, in our actual simulations, the resolution is much higher. This is just for illustration. And the key point I want to make is that we use the gridded meteorological datasets to calculate atmospheric dispersion of each single spore, such that overall the entire ensemble follows the atmospheric flow regime. And then based on precipitation values and other variables, the model calculates a spore deposition pattern. And so this is the gray thing appearing here. and these deposition patterns tell us what mass of spores is likely to be deposited. And that is the key raw result of the model, which we then analyze further, compare to data sets, and also feed into other model components. We tested the modeling environment in a previous case study with datasets from Ethiopia. Now today I'm going to present results of a new study on the identification of key long-distance routes. Here you see again the area of interest with the wheat-producing regions. Now the first step of our study is that we identify a set of key potential stem rust source locations in close collaboration with Dr. Hodson. These black dots represent all the source locations we consider, and then we additionally consider national wheat growing periods in all the countries of the domain of interest. And then we simulate a daily spore release during the consecutive wheat seasons over 12 years, and we sample daily deposition patterns on a 10 times 10 kilometer grid. Sorry, I forgot that. Now to clarify that a bit, let me show you a series of consecutive deposition patterns resulting from daily release of 1 million spores from the source location in Ethiopia. So this black dot is the source location for which I showed you the animation of the single particles. Now we'll look at a sequence of daily deposition patterns from July until December 2014. So the small map here shows deposition on the regional scale where the vast majority of spores are being deposited. And the large map also shows the tail of the deposition patterns. Now it's important to note that on these large scales, only very small proportions of spores are being deposited. But in case of a local epidemic outbreak, huge numbers of spores of the order of 10 to the power of 11 spores per day per hectare are released. So in that case, we do have to consider the tails of the deposition pattern if we want to assess probability of long-distance transport. Now, one interesting thing about that animation is that it shows the huge temporal and spatial variability of these deposition patterns that we get. For the identification of long-distance airborne routes, we're now interested specifically in spore deposition from a source location to the wheat-producing regions in a receptor country. So to clarify that, one example route here, Ethiopia to Yemen. What we do is we step through all the daily deposition patterns we get from the 12-year study period, and then we analyze on how many days do we see deposition occurring on the wheat-producing regions. We also analyze the mass of spores being deposited and specifically the relative proportion of the total deposition being deposited on the wheat-producing regions. Now in this talk, I'm going to focus only on deposition days, but in order to understand the model, it's important to note that we really are looking at the relative probabilities of these deposition events. Following these steps, we analyze the routes from every source location to all receptor countries. So for example, for the source location in Ethiopia, we do not only look at the route to Yemen, but we also look at the route from Ethiopia to Sudan, to Kenya, to Uganda, to Iran, to all the countries, and we do that for all the source locations. So we end up with a large number of deposition routes, and I'm now going to step you through a selection of key results. Hereby, I'm going to start with the geographic focus on South and East Africa, and then we'll go to the transition between the Rift Valley zone and West Asia, and then finally we look at potential deposition routes from West Asia and from East Africa to South Asia. So let us start with airborne transmission routes, likely airborne transmission routes in Southeast Africa. If we first only look at the most frequent transmission routes which we find, then the following picture emerges. These red arrows show those transition routes on which we find deposition events occurring on more than 70 days per year, on average over the 12-year study period. And first, from a broad perspective, we see that there's only very infrequent dispersal events between South Africa and East Africa. So our model predicts the separation of these zones, which is in accordance with the notion of the Rift Valley epidemiological zone, and then the South African epidemiological zone. However, we do see some deposition occurring, but very infrequently. Now, if we focus on the South African zone, we see a very frequent route from Zimbabwe to South Africa. And further up north, we see a whole set of very frequent routes in the Rift Valley zone. Here, it's interesting to point out, I think, that we see more frequent dispersal from Yemen into the Rift Valley, so into Ethiopia and Eritrea. And also, we see very frequent dispersal from Ethiopia southwards to Uganda and Kenya. However, if we additionally consider all the routes where we see deposition occurring on 21 to 70 days per year, we see that really the entire Rift Valley zone is a region of high frequency airborne dispersal. Now, this is in accordance with the generally accepted paradigm of the Rift Valley zone as a region of high inoculum airborne exchange. But here we can go beyond that and give estimates of typical frequencies of these routes and also of typical amounts and of the relative probabilities between these different routes. And we can also go and look at specific cases. So, for example, I'd like to point out that model results indicate that Uganda predominantly acts as a spore receptor country. And another interesting detail is that the most frequent dispersal into Yemen occurs from Eritrea, suggesting Eritrea as a possible source of incursion of novel strains into Yemen. Now, instead of going further into details, I'd like to highlight another key overall result. So far, I've shown you annual mean values of deposition. However, if we look at deposition events only during a specific month of the year, then we find very consistent seasonal deposition trends. So if we only look at one example route here from Kenya to Ethiopia, then this histogram here shows the mean number of deposition days in all the months of the year. And we see that in August, in case of a local epidemic in Kenya, we would expect spore dispersal actually happening on nearly every day of the month, whereas in November, any spore dispersal event is highly unlikely. This can be explained by taking into consideration dominant meteorological circulation patterns, and we find these seasonal trends for many of the routes we looked at. Now, due to timing constraints, I can't go to details here, but let us instead switch geographic focus and now look at the transition between the Rift Valley and West Asia. So in particular, we now look at airborne transmission routes between the West Asian countries and Egypt, Lebanon, Turkey, and Iran. Here, overall, we find that on the majority of routes, there's no or only very infrequent transmission happening. However, there are a couple of exceptions, and the most likely route of incursion from the Rift Valley zone into West Asia is the route from Yemen to Iran. Note that we find deposition occurring only on 5 to 20 days per year, so infrequently compared to the Rift Valley transmission routes we've looked at. This route is, of course, interesting as the UG99 TTKSK race was detected in Yemen in 2006 and then in Iran in 2007, so model results support the hypothesis of airborne transmission here. Now, the other way round from the West Asian countries to the Rift Valley, model results indicate that the most likely route of incursion is the route from Egypt to Sudan with a seasonal peak in March, and then we find an alternative route from South Iran to Yemen. As the last geographic focus, I'd like to look at potential dispersal events from West Asia to South Asia and from the East African countries to South Asia. If we start in Iran, then model results indicate that there's the potential for frequent stepwise spore dispersal from Iran to Afghanistan, and then from Afghanistan going on to Pakistan and India. So in case of a local epidemic outbreak, model results indicate spore deposition events occurring on 70 days per year, and then going on less frequently, whereas direct transmission from Iran to Pakistan is only happening infrequently. If we now look at possible direct incursions from the East African countries to South Asia, then we note that we don't find any deposition events from any of the East African countries. However, we do find the potential for infrequent dispersion events from West and East Yemen into Afghanistan, Pakistan, and India. Now, looking at this picture here, it's really of utmost importance that we only consider dispersal events. So we're not considering environmental suitability for infection yet. So even in case of a local epidemic outbreak in Iran or Yemen, when model results indicate the potential for airborne dispersal, if environmental suitability for infection is low, then of course the risk of spread is low. So we're not trying to construct risk scenarios here. We're simply only looking at airborne dispersal events. And with regards to the airborne dispersal, we think that we do make some progress in terms of a quantitative understanding of the airborne transmission routes. And we hope that we can contribute a bit to sampling strategies. For example, in case of a local epidemic outbreak, we can identify likely routes of long-distance dispersal, and we can identify risk months based on the seasonal deposition trends, and we hope that might be helpful. Now, in terms of future steps, I would like to shortly outline key future steps of our work. Primary focus at the moment and during the next months is on coupling the dispersion models with environmental and epidemiological submodels to assess the risk of disease bulk up on the ground. Next key step is continuing our work on seasonal forecasting of spore dispersal. So I briefly mentioned that we find these very consistent seasonal deposition trends, and here you see a set of maps illustrating the consistency of these trends. So it's deposition patterns from Ethiopia in November over multiple years, and we see that deposition predominantly occurs towards the southwest. And we find these patterns for many of the source locations we looked at, and we hope to be able to combine them with seasonal meteorological forecasting of environmental suitability so that we can assess risks of disease outbreaks ahead of the season, or at least approximate it. Now, in terms of moving toward an effective early warning system for stem rust, one of the key fields of ongoing work and of future work is to continue the work on a test version of a short-term spore dispersal system that we're running at the moment. So during the ongoing wheat season in Ethiopia, we run daily real-time simulations of spore dispersal from the latest detection sites in the field. So this map here shows an example of the results from yesterday, indicating likely deposition of spores tomorrow. And we see Ethiopia here. The gray areas are wheat-producing regions. The green dots are cities. And then the essential information, the gray dots are detection sites where during the last weeks, trace amounts of stem rust have been detected in the field. And we use the short-term forecasting meteorological data from the UK Meteorological Office and feed them into our dispersion model to calculate likely deposition of spores from these detection sites. Now, at the moment, this is really just a proof of concept, but we hope, as Dave Hodson mentioned, that we can extend that in the future by using crowdsourced survey data to update the source locations, and then specifically by coupling in environmental and epidemiological models so that we can provide these short-term forecasting maps in terms of risk and hazard maps and not only in terms of these deposition patterns we're looking at the moment. With that, I would like to thank you for your attention, and thanks for the opportunity to speak.